We'll be talking about does molecular genetics support human evolution. Here's the layout of what we're doing tonight. 20 minutes presentation offered to both sides. Dr. Charles Jackson on the creation side, Abby Smith on the evolution side. And we flipped a coin, actually, to decide who would go first. And Dr. Jackson's going to be going first. I don't know if it's better to go first or to be last, but 20 minutes presentation, 20 minutes presentation, 10-minute rebuttal, 10-minute rebuttal, and then open for questions from you. So we're just going to begin. Dr. Jackson is a scientist with Creation Truth, and Abby is a research student at OU Health Science Center. So we have two very qualified people that are going to be talking about the evidence for molecular evolution, okay? So we welcome them. Let's give a great big welcome, and we'll let Dr. Jackson begin.
Yeah, so tell me when the time is being kept. Are you keeping time now? Oh, okay. Well, as you might know, this stemmed from some letters to the editor of the local newspaper, bandying some he said, she said stories back and forth. And now we're going to actually do some he said, she said scripted instead of just stories. And it's, of course, about does molecular genetics support human evolution? And most people have heard of the DNA molecule. If that sounds like a big word to you there, we're talking about DNA molecule, we're talking about chromosomes, we're talking about genes and genetic sequence. This is Richard Feynman. He was played by Matthew Broderick in the movie Infinity. He was the scientist who discovered how come the space shuttle blew up in the 1980s, and he says this about science, very astute saying. Science is a long history of learning how not to fool ourselves. And I think anyone working in science has got to watch out for that. I think anyone going in any kind of an endeavor that has any wiggle room for what's really happening and what's not needs to watch out for fooling ourselves. This is Dr. Milford Waltliff from the University of Michigan, Ann Arbor, and he said this in the book Paleoanthropology. I believe a philosophical framework is not something that can be eliminated in order to provide objectivity. In my view, objectivity does not exist in science. Even in the act of gathering data, decisions about what data to record and what data to ignore reflect the philosophical framework of the scientist. If anyone tells you they're 100% objective, they're lying, at least to themselves. I'm not. I interpret all new scientific data through the creationary lens, the creation paradigm. But we have to watch ourselves with these things that we have. Now, I attended the talk by Richard Dawkins at the Field House a couple weeks ago, and one of the things he said, coming from his paradigm, was science answers the how question. The why question is just a silly question. The question is an inappropriate one. So here a question is being labeled inappropriate by a scientist. Also attended when Michael Roos spoke at the Music Building. He said about intelligent design, I'm not saying that it's a bad answer, I'm saying that it's not a scientific one. Some answers just aren't appropriate. So in certain paradigms, some questions are appropriate to ask, some answers, though they might not be bad ones, are still inappropriate to offer. These things are ways that we can fool ourselves. Does molecular genetics support human evolution? We're talking about DNA, we're talking about DNA sequences, and we're talking about the evolution of human beings. This is a chart showing, yes, different kinds of apes and then the human here. We don't disagree about what's above the tip of the iceberg. What we disagree about is how we all got here. Sure, chimps are very similar to us and the great apes, but how did we get here? Two different stories. Many evolutionary scientists have recently expressed doubts about the validity of molecular genetics evidences for evolution. This is Dr. Waltliff again. He's in Science News magazine this time in 2004. How many of you ever heard of mitochondrial DNA or mitochondrial Eve? Okay, so you have some familiarity with what they're talking about there. Those who use mitochondrial DNA or the mitochondrial clock to reconstruct animals' evolutionary histories assume that its chemical sequence changes only at random, but mounting evidence indicates that natural selection molds the makeup of mitochondrial DNA, making such analyses useless because the clocks really don't run evenly. Also, I attended the lecture of Dr. Solis, who spoke at the Sam Noble Museum just a short while ago also, and she said on the molecular DNA clock, she said we know that it doesn't tick evenly, regularly, or the same in all species, and you wouldn't trust a watch like that either. I'm not saying that they're totally off on this, but I am saying that some people are just vesting in hopeful thinking. Now, a lot of times evolutionists like to point out that humans and chimpanzees are so very, very much alike. And it's true. We are more physiologically and structurally the same to chimpanzees than anything else. Out of 3 billion base pairs, 1.23% is the only difference. That's still 36 million base pairs, but we still have, 98 some odd percent the same DNA as chimpanzees. Well, they look the most like us of anything. It's not so surprising that structure and genetic coding would be the similar. But here's a nematode worm, microscopic, only has 1,000 cells in its whole body, and this we have a 75% similarity with in our DNA. All living things have similar DNA, some more than others. Now, here's the kicker, bananas share 50%, the same DNA as we do. Now, I'm not making this kind of stuff up. All living things do. Now, if those similarities between very, very different kinds of organisms compared to us exist, then it's certainly not surprising at all. And doesn't necessarily mean that we evolved from monkeys. If they were created the same time as humans, and they look a lot like humans, it's not surprising that their DNA would be very similar to humans too. So that issue is transparent to the entire thing about evolution and creation, and whether humans evolved from chimpanzees or not. Now, I wanted to emphasize that our DNA really is very similar. If you look at this here, here's a karyotype of human, chimp, gorilla, and orangutan chromosome number 12, 13, and human chromosome number 2. And look what it says here. Of these 4 species, chromosomes 6, 13, 19, 21, 22, and X have identical banding patterns. When you look at them through the microscope, you can see that these look almost like carbon copies of each other. Chromosomes 3, 11, 14, 15, 18, 20, and Y look the same in 3 of the 4 species, those being the gorilla, chimp, and humans. And chromosomes 1, 2P and 2Q, which is the human 2, and the chimp 12 and 13. 5, 7, 8, 9, 10, and 12, and 16 are alike in 2 species. Only chromosomes 4 and 17 are different among all of them. The biggest single chromosomal rearrangement for the 4 species is the unique number of chromosomes, 23 pairs, found in humans as opposed to apes. Now, this is something evolutionary scientists are fond of pointing out, that chimps have 24 pairs, humans have 23 pairs, and that second human chromosome looks an awful lot like the combination of chimp 12 and 13. Well, indeed it does. Now, what they're saying is that chimps and humans were hanging around, and then these two fused, and look at how similar they are. And now in humans, we got this chromosome 2, which used to be chimp 12 and 13. Now, I have no problem, as I just said, with the chromosomes being very, very similar in chimps and humans in the past, since we look alike anyway. But what evolutionists will say is that what happened was, back in the day when we had a similar number of chromosomes, these things here fused, made one chromosome. That's human chromosome number 2. It's a combination of 12 and 13 from the chimps. There it is now, and it's all beautifully laid out. You can just see that proves evolution. It doesn't. The similarities actually could go easily either way for design or random evolution. The fact that after the creation or the evolution of these 2 species, that the humans fused chromosome 12 and 13 is not a similarity. It's something that happens that shows the species are separate. It doesn't show they were the same, or chimps that have the fusion too. So my story is that yes, humans and chimps did once have the same number of chromosomes. Humans had a fusion, but it never did happen. In the chimp line, it's an evidence of not being related, not of being related. So again, like I said, it's not the tip of the iceberg we're arguing about. It's how the iceberg got the way it is. What I'm saying is that there isn't evidence for a common ancestor as far as molecular genetics goes. DNA studies have forced the rejection of two of the great missing links that were in the evolution story, Homo erectus, or upright walking man, and Sahelanthropus chilensis, the Tamai child. Here we see in Newsweek magazine in 2007, DNA makes clear that Homo erectus was almost certainly a dead end and not, as some scientists had argued, our ancestor. Trouble was, Sahelanthropus chilensis, nicknamed Tamai, the word for child, lived close to 7 million years ago. The genetic data, pointing to a chimp-human split at least 1 million years later, suggests Tamai is not the hominid, not the first creature ancestral, only humans, not our chimp cousins after all. So that knocks two of the members off the evolutionary family tree, and the other ones have their problems too, but these are the two that had the big problems with the molecular genetics. Now this textbook here, very popular high school biology textbook, does have these two species as missing links. Here you've got the Sahelanthropus chilensis, recent hominid discovery. The book was published in 2006. Here you have Homo erectus, also mentioned as a missing link to humans, and yet the Washington Post knows that these were not human ancestors. The book even has a little part where the students can pick which of the stories of evolution they think is really true. The only difference is Australopithecus africanus is not listed in this one, but Homo erectus is in both of them, and of course, as we just found out, Homo erectus is, because of the DNA, not a human ancestor. DNA studies have forced the acceptance of unlikely separate evolution of specific traits. It turns out the way that evolutionary paradigm scientists interpret the DNA structure of bats, for instance, forces them to say that echolocation evolved at least twice on two separate occasions, as much of a lightning strike hopeful thing that was to happen just once. Also, DNA studies have forced the acceptance of repeated evolution, then de-evolution of specific traits. And this goes against evolutionary principles. This is the Phasmagigas, a foot-long walking stick insect, and it says here in Science News that stick insects may have done what biologists once thought was impossible, lose something as complicated as a wing in the course of evolution, but recover it millions of years later. Says Brigham, let's see, that's not supposed to happen with so-called complex traits, at least according to a long-reigning view of evolution, which is Darwin's principle, says Michael Whitting of Brigham Young University. Yet that's the story revealed by the new family tree based on DNA data from 37 species of stick insects reported in 2003 Nature magazine. At the top of the DNA tree, wings appeared, seeming to have re-evolved at least 4 times on 4 different occasions. The wings popped back into the scene via accidental evolution after up to 50 million years of winglessness. And it's the DNA studies. It's not like if you were an evolutionary theorist, an evolutionary mechanic, a theorist, that you would want to say these kind of things or want to see these kind of things. It's where their way of interpreting DNA has led them and must lead them. Again, Dr. Feynman saying science has a long history of learning how not to fool ourselves. I'm finding evolutionists are moving away from hard data, going to more numerical data, going to more highly complex mathematical models, rather than actual hard science. I did attend a lecture of John Beatty, also recently here at OU, and he said, if outcomes are chance, then maybe the laws are chance too. The laws of evolution could have turned out differently. And once they say the laws evolved, we'll realize those aren't the laws. The laws of nature, thought, comes from a time when science and religion were closer. We might have moved beyond the laws of nature. And of course, if a paradigm moves beyond established laws of nature and says it is above those laws, well then of course you can say anything. And then I do think that we are not heeding Richard Feynman's admonition to not fool ourselves. So that's what I had to say about molecular genetics and not supporting human evolution. Thank you.
Okay, can you you guys hear me okay? Awesome. I'd really like to thank the pastor for arranging this for us. I really don't get a lot of opportunities to have these kinds of science outreach opportunities. And I'd really like to because I know in Oklahoma there's a lot of contention between evolution and creation. And I think part of that is a lot of the general public doesn't understand why scientists get so worked up about evolution. Because, I mean, to the general public, it could be like an amoeba evolving into a frog, evolving into a chicken, evolving into people millions of years ago. Why is this such a big deal? Well, I use evolution every day in the laboratory in two ways. The first, I think some of you would call in a microevolutionary sense. I study the evolution of HIV in the hopes of creating a vaccine that will eliminate that epidemic from this planet. And I also study it in a different way. Some of you might call it the macroevolutionary sense. And I do that in hopes of curing cancer and preventing cancer. So this isn't just about abstract things that happened millions and millions of years ago. It's a tool that scientists need in the lab to help people.
So what do I mean when I say evolution?
Okay, all I mean is descent with modification. So you're different from your parents, and they're different from their parents, and so on and so on and so on. That's it. If you carry that back far enough, you come to the exact same conclusion as Charles Darwin did 150 years ago. And that is that all organic beings have descended from one primordial form. And this was 150 years ago when we didn't have the technology that we have today. And he had this really cool drawing in one of his notebooks that actually recapitulates what we do in the laboratory when we're comparing one organism's genetic code to another organism's genetic code. So his insight really was astounding for the time. But how do we make trees like this? Like Mr. Jackson said, we can compare things that we have in common with other organisms. One of those things is the PAX6 gene. So over the course of time, as the eye was evolving, this particular regulatory gene can be found in people, in mice, in birds, in fish, in insects, to the point where you can actually take the PAX6 gene out of a mouse, put it into the fly's genome, and you get a normal fly with a normal eye. So we have things in common with other organisms. So to a scientist, we look at this and think, well, we had a common ancestor, and that gene got passed on from generation to generation to generation. But, like Mr. Jackson said, does that really support common descent? Because if you have a toddler playing with Legos, she can make a spaceship from that set of Legos. She can make a castle. She can make a dinosaur. If she's using the same building blocks to create different things, I mean, that doesn't mean that the castle is related to the spaceship. What really tells scientists that we are related to other organisms are these weird mistakes we have in common with other organisms. These mistakes don't make sense in light of any other paradigm than that of descent with modification from a common ancestor. One of these mistakes is what I study in the lab. They're called endogenous retroviruses. And I contend that if Darwin was never born, or if he was born and he decided to open a salon in day spa in Cuba or something, and every fossil remained buried, if we managed to figure out how to sequence DNA, the second that we could compare genomes to one another, and we saw the way these endogenous retrovirus patterns set up in all the organisms, then descent with modification would have smacked us over the head like a pillowcase full of doorknobs. So what am I talking about when I say endogenous retroviruses? Well, everybody has heard of retroviruses, of course, like HIV. Unlike humans, which we carry our genetic information as DNA, which gets turned into a message RNA that gets turned into protein, retroviruses do things a little different. They go from a genome that's RNA, and then they turn it into DNA, and they sneak that into our genome. And so what happens is this virus becomes a permanent staple of that cell's genome. And it tricks it into thinking, oh yeah, this is a normal cellular protein. I'm supposed to be making this. Yeah, everything's fine. And they end up making all these viruses that go on to infect more cells and wreak the havoc that we know of. Now, endogenous retroviruses, they're a mistake. It's what happens when a retrovirus infects a cell that it's not supposed to infect, like a sperm or an egg. Normally, they want to infect your immune cells or epithelial cells or something like that. But when they infect one of these sperm or egg, if they go on to participate in fertilization and that embryo doesn't die from the genetic mayhem that that virus causes, then what happens is that permanent resident in that embryo's genome, that permanent retrovirus, gets passed on like any other gene, just like the PAX6 gene, just like the gene that says, what color that embryo's hair will be. It's absolutely normal. So, when we look at the human genome, and we look for endogenous retroviruses, we see that some of them are unique to humans. We can't find them in other species. But some of them we have in common with other primates, with chimps, with gorillas, orangutans, with gibbons, with other old world monkeys, going all the way back to new world monkeys. But these aren't just static landmarks that scientists stated, oh yeah, evolution happened because of these guys. These endogenous retroviruses, it's a very unlikely event that one of these events happens, that a retrovirus infects a sperm or an egg, that goes on to be an embryo, that goes on to be a normal organism. But when we're talking about millions and millions and millions of years, random unlikely events happen a lot, to the point where about 8.5% of your genome are these endogenous retroviruses. You're 8.5% viral, which I think is cool. And so you've got tens of thousands of these retroviral genes in your genome. On the bright side, most of these are non-functional. They've accumulated mutations, they've accumulated deletions and frame shifts. They don't work anymore. That's really good. But unfortunately, there is a few dozen that can still code for a protein. Now, cells still have ways of keeping these guys quiet, but sometimes that doesn't work out. And what we found is that in basically every cancer we look at, we can find these endogenous retroviral particles coming out. So when you've got these retroviruses coming out, popping in, causing a lot of genomic instability, which leads to cancer. And that's why we don't know whether it's a cause of cancer or whether it's an effect that perpetuates cancer. But we need to understand these endogenous retroviruses and their place in evolution to just figure out what's going on so we can hopefully figure out a cure for these diseases. And it's not just cancer. They're associated with multiple sclerosis and lupus and a lot of other diseases. So to scientists, this isn't just an abstract thing that, oh, we're just obsessed with a paradigm and we can't look at things any other way. It's really important to us. Unfortunately, the creationist response I've had to endogenous retroviruses is baffling. The Institute for Creation Research just says that endogenous retroviruses don't exist. When they speak about endogenous retroviruses, they say supposed or so-called or whatever. They're not real. Intelligent design creationists think that scientists are making them up. Like, we're looking at this DNA code and like a beautiful mind, we start seeing all these patterns coming up of viruses. No. Viruses, I think a lot of the general public think are like these alien, kind of foreign creatures. They're actually a very diverse species, families. So HIV is as related to the youngest retrovirus in your genome as we're related to green algae. So there's a lot of diversity in retroviruses. So when we say there's a retrovirus there, we know what we're talking about. Sometimes they say that retroviral insertion isn't random. And that's absolutely a valid question, because we know that humans and chimpanzees can be infected by the same retroviruses. That's how we got HIV. But again, they're forgetting the diversity of retroviruses and the randomness of where they insert in genomes. Just recently, we found a very unlikely retrovirus in lemurs, a whole different species of lemurs. And so scientists thought, well, maybe this retrovirus inserted in a common ancestor of lemurs like 85 million years ago. And that didn't seem quite right to them, but it's a possibility. And they also thought it was a possibility that this retrovirus independently inserted in several species of lemur. So they didn't just guess or throw up their arms and say, whatever, it's the older one. They looked at those sequences and determined that they were actually very different retroviruses, and that these were two independent events. So we know when things are random, and whether it's common descent that is the more likely explanation, or independent insertion sites. Sometimes they say ERVs don't work because not every organism has them. That's true. Plants don't have endogenous retroviruses, but they have things that are very, very similar. And so we can work back phylogenetic trees of plants using those weird mistakes. Because ERVs, they aren't the whole picture, they're just a part of the picture. And ERVs paint a big portion of it. And worst of all, some say that 8.5% of your genome that's retroviral is not really a mistake, they're all functional. And where they get this idea is that it's very, very cool. But we have mammals because an endogenous retroviral protein was domesticated by your genome. So this retrovirus inserted, and instead of just having it delete itself into nothing, your cell was like, hey, this is useful for something. And so all mammals actually used an endogenous retroviral protein to create a placenta. We exist because of a viral protein. I think that's awesome. The problem is, creationists take that to the extreme and say, well, because this one is functional, they all must be functional. They're not all functional. We know what happens when you're infected with one functional retrovirus, AIDS. And so you don't want 8.5% of your genome making all these viruses. You get cancer, you get all sorts of diseases. It's a good thing these guys are junk. So why do I care personally about evolution in classrooms and that sort of thing for kids? Well, because I'm very confident in the education I'm getting here in Oklahoma. I love it. But I'm not vain enough to think that I'm going to cure AIDS and cancer personally. So I'd really like to use my career to advance scientific knowledge in my field, and then have kids come up after me to be graduate students when I'm a principal investigator, to take over the labs when I want to retire someday. So that's why I care, and this is how we use evolution.
Okay, I didn't know people could be so short-winded. Yeah, they both were short on their time, so we have a lot of time ahead of us. So we still have 10 minutes for rebuttal of each one. So, Dr. Jackson. Okay.
Well, let's see. Well, we do agree on a number of things, the similarities between lots of species and their DNA, and also on some of the similarities between mistakes. One thing I will point out, a point that was made, is that descent with modification is what evolution is. If that's all, just that I look a little different from my great-grandfather, then I believe in evolution. But you see, the difference was also drawn between microevolution and macroevolution. In the creation paradigm, there is a limit to how much change can happen just by reshuffling the genes that are already there. By evolution, new genes must be created, coding for new traits with new information, and thereby creating, as Darwin said in the title of his book, the origin of new species. Well, there are limits to the changes, and I'm going to show you that in just a second. We share these mistakes, these endogenous retroviral sequences in our DNA, 8.5% of our DNA being made of these things, and that this is some kind of a marker that shows that, yes, we're related to chimps. It does make an assumption, And I'm not saying assumptions are bad. In science, you must make assumptions when you don't know something. The assumption is made that the only way to really get these things into the genome and into the germ cell lines is by common descent. And that two separate species that never were separate shouldn't be showing the same endogenous retrovirus DNA sticking there in their chromosomes. Well, how many of these are actually orthologous? How many of these Earths actually are the same ones and are on the same location? And what are the chances of being able to happen if there's 98,000 of these that exist in us, and however many in chimps, maybe 200,000 of them exist in us? Let's take a look at something from an article from last year, from the Proceedings of the National Academy of Sciences. It says that these things could go back 85 million years, according to DNA clock testing that evolutionists use. Lentiviruses could be very ancient indeed, 85 million years. Now, viruses are known to mutate at an incredibly rapid rate, maybe 1,000 times, maybe 1,000,000 times faster than other creatures' DNA, like yours. Now, if these viruses have been multiplying and mutating at these incredibly accelerated rates for what this article claims is 85 million years, well, 1,000,000 times that is 8,500 trillion years. And that's in people years. That's different from dog years and people years. I'm talking about people years and virus years, okay? They've had the equivalent of 8,500 trillion years, and what are these lentiviruses? What are these endogenous retroviruses today? They are still viruses. They haven't written any new code with new traits or new genetic information. They haven't advanced any. They've done exactly what the creationist paradigm said. By shuffling genes and by innovation of genes by accident, they can make horizontal changes, but they can never get past that limit to change that I was talking about. Until now, scientists thought lentiviruses, like HIV, were too young to have participated in this evolution back and forth. Well, just this past week, I think this is probably the one that we were just referring to, the two different lemur species in Madagascar that were found to have the same, and it's very clear, the two different species of lemurs in Madagascar suffered independently and just about the same time multiple germline infections. It says here that the whole thing was older than previously inferred by the DNA differences between between what they see from the past in these now existing lemurs and what they see in living viruses. And that happened independently. Two separate lineages got this endogenous retrovirus. Well, viruses can infect different species. It happens all the time. The report here in Public Library of Science Genetics just of this week says the viruses that were endogenized or got into the genome in the two lemur species were nearly identical. Yet, of course, these might have been 38 million years ago, but today the sequences are nearly identical, showing that they are probably the same thing. Two different species of lemurs got the same cold or whatever it was. You know, isn't it possible that people and lemurs or people and chimpanzees could get the same virus at the same time, and then those same endogenous retrovirus sequences can run into the genome, and it can, yes, indeed, make it look like, if your paradigm is that chimps and humans have the same grandfather, it would look like that would be in support of that. Or it could happen, it could fit right in with the creation paradigm too. They both just simply contracted the same virus, which then implanted the retrovirus sequence into the genome. Again, this is transparent to the issue. How many of these things are actually at the same location, on the same chromosomes? How many of them are, in other words, orthologous? They really are, or homologous. And how similar are they? I think this probably needs more research. I think you will see evidence that goes both ways easily in molecular genetics. I think you will see evidence that goes one way or the other. Personally, I think you'll see more evidence going the creation paradigm way, but evidence is not proof. It's stuff that helps you think that your opinion is stronger, but proof is what's needed in science. Data, logic, reason, and thinking. That's what we need. It's okay to have theories. It's okay to make assumptions. Those things are not wrong, but we should not teach them as though they're facts. And here's just a list of other viruses that animals and humans both can contract. And you can certainly have these viral DNA pieces, sprigs, in both humans and animals of any of these in modern times. Thank you.
Just a little clarification. When a retroviruses mutate really fast. That's because when they are converted from RNA to DNA, the protein that does that doesn't have any proofreading mechanisms like our cells have. So they end up making a lot of mistakes. And that's actually one of the points where mathematical models have really, really helped, because you get so many variations with HIV that it's almost impossible to test them all in the lab. So math is also our friend. It's not a cop-out. But once it becomes an endogenous retrovirus, it has the exact same proofreading mechanisms as our own cells have, which is actually pretty good. And also about getting new genes and new information. I'm going to talk about that just a little bit. But if you're really interested in how viruses can create new genes, Google Abby Smith and HIV, because Michael Behe, an intelligent design creationist, made the claim that HIV is the super mutator and it can't get any new information, when in fact it kind of has. So Google that if you're interested. But I want to talk about how our genomes get new DNA, because it's actually a really cool story. And I want to dismiss the kind of pop media culture claim about junk DNA while I have everybody's attention here. So when you hear a science reporter say something about junk DNA, it's always along the lines, scientists thought this was useless and now it's got to function. That's not what junk DNA is. Junk DNA actually has a very specific definition that came up in the 1970s, and it's a very cool story I'm going to tell y'all. So what happened was, back in the 20s, 30s, 40s, 50s, long, long ago, before we had all this really cool technology, scientists were just figuring out that DNA is how inheritance is transmitted from parent to offspring. We didn't know that back then. And so they thought that these big, elegant, complex creatures like ourselves must have a ton of DNA, whereas we'd have more than, say, a dog, which would have more than a chicken, which would have more than a fish or a turnip. So they started seeing all these sizes of the DNA they were finding, and a species of onion has twice as much DNA as we have. And a different species of onion has even more, and a different species has almost 10 times as much DNA as we have. And that's kind of degrading to think that this onion has all this DNA. Why don't we have all this DNA? So Susumu Ono thought of this really cool idea, which has actually become a staple of basic genetics. And he thought, since the fossil record was scattered with all of these fossil remains of extinct species, maybe our genomes accumulate all these fossils of extinct genes. And so what he did was come up with the idea of gene duplication and divergence and pseudogenes. So gene duplication and divergence is one way that we get new information into our DNA. So for an analogy, let's say you just graduated from high school, and from like some graduation party, you won a free computer. Awesome. You get home, grandma just got you a free computer. She bought it a while back. You can't take it back. You don't just want to sell it on Craigslist for $500. So you decide that you're going to make one of these machines, you're going to have it evolve into your gaming machine. So you get a sweet game, you get World of Warcraft, get a new monitor, get a new graphics card, gaming mouse. That's your gaming machine. Your other computer, you say, is going to be for school and research only. It's going to be your professional computer. So you keep all your data there, you get an external hard drive, nice professional case to create in, maybe some presentation materials. So that's kind of what happens with your genome when you get a duplication and divergence.
And this is what has happened with
The evolution of the immune system. So you have a particular kind of antibody called IgM. Really cool shape. It's the first one that's made. Neat. It performs whatever function you could possibly think of. But we don't just have this one kind of antibody. You have a ton of different kinds of antibodies that have been the result of all these gene duplications. And so you've got 6, 7 different antibodies that have been able to evolve in their own particular way. You have one that's functional, and one that gets to kind of explore sequence space to see if it can do something different. And that's precisely what happens. So IgA, for instance, is really good for mothers who are breastfeeding their infants. It's really good to breastfeed because infants don't have a really good adaptive immune response yet, so they get antibodies from mom. The IgGs are really good at attacking bacteria or anything that is invading your immune system or invading your body. Whereas IgE, I don't know why this evolved. It's the reason we have allergies. I never had allergies, so I moved to Oklahoma. I'm not a fan of IgE. So where do we get these pseudogenes and junk DNA, what Tsusumu was talking about? Yay, 2D computers. Ooh, you drop one and smash the screen. It is now what you might call a pseudocomputer. You can take some parts from it, you know, if maybe you spill pop on your keyboard, you can take the keyboard out of your pseudo and maybe compensate. But it's not functional as a computer anymore. It's a pseudocomputer. So to go back to ERVs, this is what happens to endogenous retroviruses. They turn into junk. That's good. Junk DNA in the form of endogenous retroviruses is great. So let's say in this particular GAG protein, GAG codes for the structural core of a retrovirus. In this one, we've got a frame shift, so we got a little insertion. The code doesn't make sense anymore. Polymerase codes for all the enzymes retrovirus needs. This one's got a big deletion in it. And the envelope gene codes for the little spikes on the outside that the viruses use to get to your cell. This one's got a whole bunch of stop codons in it. So it's not functional anymore. It's junk. It's awesome. You don't want these guys functional. But that's just one of the ways that you accumulate information in your genome. And even like these leftover bits of junk can still be used for other things. Like you can take the keyboard out of that junk computer and use it for something else. So these little LTRs, those are the promoters for the virus saying, like, hey, come here, make these genes. They work really, really good. So sometimes your body's like, wait, this one works a lot better than the promoter I have. I'm going to get rid of my old promoter, use this one. So that's how you get new information.